Good morning, good afternoon, good evening, good day, good night. Welcome. This is the first of 3 videos associated with your second InvertebrateR or InvertebrateR assignments. And we're going to talk in this one, we're going to continue our exploration of the deep sea hydrothermal vent community, this endemic, species-rich, gorgeous, and recently discovered, relatively, like to an old person like me, relatively recently discovered community around the world. And we're going to be extending our exploration from the assignment 1, where we focused on a set of vents near Australia to look at dispersal. We were kind of building on what we'd learned about plankton in the lecture, about planktotrophic and lectotrophic, and we're adding to that a discussion of metapopulations, patchy populations protected by a not all your eggs in one basket kind of a scenario, and linked by dispersing individuals. And how do we measure dispersal? Well, indirectly we can do it using genetics, and sometimes using one method of doing that is using tree structure. And so you did that, so well done you. And we're going to expand that to build on consequences of dispersal limitations. One of the consequences of dispersal limitations is, of course, isolation of any one of these populations. And so one measure we have of isolation from an ecological perspective is beta diversity, or dissimilarity. How similar or dissimilar are two communities, two places for the species that live there. And we can also measure alpha diversity. How species rich are they? If we're going to apply value to one system or the other, or one place or the other, perhaps it's the alpha diversity that we're going to use to do that. So we're going to measure alpha diversity. We're going to measure beta diversity for deep sea hydrothermal vent regions around the world. And we're going to ask how unique they are and what consequences are associated with mining. And so we're going to today, in this spiel, we're going to reintroduce you to the thermal vents. We're going to reintroduce you to the scenario, the kind of the envelope scenario of what do we do when human enterprise wants the minerals, the rare and special minerals that are associated with the deep sea vents. We're going to give you an introduction to non-metric multidimensional scaling. That's what NMDS stands for. And then talk about the questions and how we would analyze those. And so then once you've watched this video, you can move on to the R, how to do it, and then the video, how to do it videos. Sounds good? Let's start. So you remember, of course, when we're talking about these deep sea hydrothermal vents, we're talking about the incredible discovery that Bob Ballard and others made on the off the South American coast in the mid 1970s. That there was life biology, not just like eking or kind of scratching its way out beyond the reach of the sun. It was numerous and abundant and specialized. So the discovery of Pompeii worms and mollusks with shells made of metal and incredible symbiosis and chemoautotrophs that we're only still discovering as we discover more and more of these vents on the largely unmapped bottom of the ocean. We're discovering more and more taxa associated with these vents. This is a globally distributed phenomenon, remember, but one that encompasses a very small area. It's kind of like these splits. They're kind of like a river, right? Little cracks or rivers. So a river doesn't include a whole bunch of area, but it has a whole bunch of species within it, because if you're aquatic, you can't live terrestrially, you live in the river. And just like these, they can live associated with the conditions that are associated with the deep thermal vents. And so we see incredible specialization around the world. And so this is my river analogy, that these cracks, these are the thermal vents and some of the ridges that encompass the globe. So this is a globally distributed phenomena that makes up a very small area. And you saw this statistic before, only about 50 kilometers squared for the regions that we know about globally. And what we've learned so far is that each thermal vent site is characterized by a multitude of species, and many of them, as we poke at them and begin to sample them and film them and bring them up for barcoding, for genetic analyses, etc., seem to be endemic and unique. And these ecosystems host a diversity of life, a biomass that's comparable to coral reefs or tropical rainforests. And they're incredibly threatened. We want the metals that are associated with these sulfide-rich sites, these rare earth metals. And so people around the world, nations around the world, but also different companies, so non-nations, but companies, are leasing. So you're looking at this map here. This is a global distribution of deep sea hydrothermal vents. So it's like the last map, but overlaid onto that are, in yellow is the vents, but in different colors are highlighted different regions where the ISA has issued mining exploration leases. And one critical twist on this is kind of the Wild West mentality, I'll say, of areas beyond national jurisdiction. So in the deep sea, outside of the influence of individual nations. And determining, because of the isolation and because of they sit outside of national jurisdiction, determining diversity and standardizing research effort is a big problem. And so knowing the players in the play and these ecological dramas on these hot thermal vents deep underwater is a real challenge. So here's another look at exclusive economic zones, marine protected areas, and mining licenses overlaid here onto these deep sea thermal vents. And you can see that there's not a huge interaction or Venn diagram between marine protected areas and deep sea thermal vents. So we're talking about areas that nations are very excited and keen, and companies are very excited and keen to begin to exploit. And as biologists, as citizens, I think one of our obligations is to bring information to the table regarding the environments where these exploitations might happen. Now, there's all sorts of, like I mentioned, the minerals, and in the last video we talked a lot about the nodules that are being potentially mined off the floor, not in deep sea thermal vents, but in wide abyssal zones in the Clarion-Clipperton zone in the Pacific. But there's just an incredible number of outputs, economic outputs, sociological outputs that could come from these kinds of explorations. Not just the explorations geologically, but biological explorations. And I'll give you one example from a species that we've talked about before, the Pompeii worm. And when the Pompeii worm was discovered, one of the things you know about this annelid is that it has no gut and it lives chemoautotrophically, and it provides this kind of sleeve for the bacteria to live on. And the bacteria produce a heat-resistant compound that helps the worm withstand the extreme high temperatures that it lives beside. Now, since the discovery of that bacterial compound, it's now been marketed and patented and marketed as abyssine, a chemical that acts that you can rub on your skin to protect and help recover from abrasions and sun damage. And so this, just as one example of something that's happened for the past 20 years now, even though most species in the marine environment haven't been described by science, and even though most of the deep sea has not been explored, and that first bullet is kind of two true statements, the compounds that we already derive from the known marine species of the known mapped deep sea are brought to market 4 times more often than those derived from terrestrial species. And multinational companies are by far the most prolific patenters of these deep sea genetic sequences, the ones that would have been manipulated slightly from the naturally occurring bacterial compound, which you couldn't patent, to the one that you would twist slightly but still have the same characteristic effect, and then you could patent as abyssine. And so you can see different nations have invested differentially, but so the number of predicted patents for the US and Japan outstrip much of Europe, but Europe kind of comes in second in third to the 2 leading agencies or nations right now, the US and Japan. And this is an issue. And so there's not just physical geological mining, but also there are other attributes that we may wish to extract. and we may be able to extract less destructively. We may be able to learn. This is kind of a biomimetic pathway instead of an extractive pathway, but one that helps us learn and adapt for our own use solutions that other species have already come to. And I'm going to kind of pause and just say, if you want a refresher on that, if you didn't take the time to look at the context video, go to that, watch me talking about deep sea hydrothermal vents and some of the details from the first video, and some of the mining context that's given there as well. So I'm only briefly going to remind you of the fact that we're very interested in mining at thermal vents, and there are a variety of extraction techniques that would be site appropriate. These ones are cutters like Roomba robots that have enormous consequences for the deep abyssal zones of the Clarion-Clipperton zone, where the nodules are being scooped up, because where we've seen these enormous threatening looking monsters working, and we've revisited them at those sites after 40 years, we see like no recovery. And that's in the deep abyssal zone, and we would expect kind of the same lack of recovery in the hydrothermal vents. And why is this kind of an interesting project, or an important one for us to be informed about and thinking about as invertebrate zoologists right now? Well, it partially has to do with an executive order from the American government early this spring, in the spring of 2025, that directed NOAA to fast track all permits for companies to mine the ocean floor in the waters under the jurisdiction of the United States of America, and also international waters. So essentially setting the grounds to bypass the United Nations agencies that have been set up since the 1960s and 70s to govern exploration on the deep sea of the world, essentially as a human place, not as a nation state. So this is a problem. This is a recent review of that problem that asks the question, is there a way to responsibly mine hydrothermal deep sea vents, the active ones or the ones who have become inactive? And these are two Canadian researchers that presume that set up the scenario that the dynamic nature, that they open and they close and they follow these large cracks, but where they open and close changes through time. So that ephemeral nature has led some to propose falsely that they're resilient to disturbance, because on the ecological scale, they may experience little or no disturbance and remain stable for decades in some vents. And the fauna that live there are where we've learned about them are highly endemic, and they experience extreme habitat fragmentation, and you've seen some of the consequences of that with the first InvertebrateR assignment. And so their proposal, Snellgroves and Metaxas, is that we need a holistic approach to management. And this is particularly difficult. It's an invocation that we need this, but it's difficult since most of the areas are outside of national jurisdiction. And we talked in the last InvertebrateR assignment about an address to the General Assembly of the United Nations that really started the ball rolling about establishing the ISA and bringing kind of the nations of the world together to think about the bottom of the ocean as not being for the exclusive domain of any one of the wealthier nations, but for a resource and a shared legacy for the world. And even just last week, in late September, at the last General Assembly, the 80th General Assembly of the United Nations, Liberia's President Boakai talked about the importance of deep sea mining and the critical point that we're sitting at right now when we're considering rushing ahead, as this executive order from the Americans suggests, to grant permits and mining leases. So we're threatening this environment before we even understand what lives there. And you can go and read his article. This is an article to his address that he published And I'll revisit the last bit of revisiting from the last scenario that we talked about was in unpacking the role that some Canadian, the leading role that some Canadian companies are playing as multinational corporations, but this one is based in Canada, the Metals Company, where their chief environmental agent or their chief environmental officer, Michael Clark, in an interview with the Narwhal that you can go and read there later on, talks about the fact that it says these three things in a statement. I've broken them up as bullets because I think the ultimate truth is in what the president of Liberia said in the previous slide and not here, because I think there's a disconnect here. He said we don't need to know the name of every species out there, and we don't need to know the distribution of every species, but what we do need to know is the magnitude of the impacts that we're creating. And I think one of the things that you're experiencing by actually looking and feeling and using this data is that we can't do 3 without 2 and 1. So, what's the particular analytical bent of this particular assignment? Well, we're going to talk about vent regions around the world, and we're going to compare them. So at multiple areas. And so we're going to figure out how we're comparing them being alpha, beta, or so richness and then beta diversity are measures of similarity or dissimilarity. Now, measuring alpha diversity is maybe intuitive. We can look at bars. We can just say that it's this much or this much with some amount of variation around if there's multiple vents that we've sampled within a particular region. But how do we do beta diversity? Perhaps you have not yet been exposed to different ways that we have of measuring beta diversity, and we're going to change that. We're going to use statistical ordination or multivariate statistics, but statistical ordination, which many people are suspicious of right off the top. But why do we need it? Because data and processes are often complicated. And even some of the greatest statisticians, biological statisticians, actually are Canadian. Pierre Legendre is one, and you'll see the cover of his book later on. He writes that no one wants to make ordinations, but when you have complicated multivariate data, so sites and taxa, and then different measures of the environment of abiotic variables like temperature or current or these kinds of things. So we're often desperate to make association, to find out which associations are real and which are spurious. And so the goal with this kind of an ordination is to whittle away the detail to show the process on as few axes as possible that still explain the biology. We'll talk about what those axes mean. So what is ordination? Well, because we've got, like Lucy here, we've got an abundance of things, abundance of taxa, abundance of sites, abundance of information. Ordination from the Latin, ordinatio, is the action of setting in order, the arrangement of units in some order. So Legendre wrote about this so in ecology, data sets usually include a large number of variables, so things we can measure, and they're associated, we presume, this is the question mark, with each other linearly. But the basic idea that underlies a whole slew of methods of analysis is to reduce the large number of intercorrelated variables to a small number of composite but linearly independent variables. And if each of these variables explains a large fraction or a sufficient fraction of the variation, then it's going to be useful. And Legendre, and you can go and look at different, like that's a link to your university library that includes your access to a PDF copy of this kind of big, thick, gorgeous book that will talk about a whole bunch of different popular types of ordination, like principal component analysis or correspondence analysis that use linear representation and eigenvector techniques to do these kinds of comparisons, not just with taxonomic data, but maybe with morphological data or chemical data. The red here, non-metric multidimensional scaling, that's what we're going to use, because we're not going to use all of them, we're going to use one. And we're going to use it because generally we use it because it's robust. And it's computationally accomplishable on everyone's laptops today in R. And we're doing it because it presumes a nonlinear association. And it works when a lot of assumptions are violated. And it essentially is going to build you a rank order relationship with community dissimilarity and ordination distance. I'll unpack those words as we go. So non-metric multidimensional scaling, or NMDS. What it tries to do is to represent a set of objects or location sites, sampling sites in this case, along a predetermined number of axes. So we're going to try and reduce it now to 2, so like first and second axes, while preserving the ordering relationships among them. It's not eigenvector based. We're not going to unpack the mathematics of eigenvectors. It doesn't depend on a linear set of relationships between your variables like PCA does, the last 2 conditions like PCA does. It's resilient to outliers because in NMDS, observations are essentially viewed as presences, and in the example, our video, you'll see me reduce what is proportional data just to incidence data, presence and absence data. And so while there's a whole bunch of other ways to look at this that you can expand in your training as a biologist, today we're going to use NMDS. And so when we talk about maintaining the rank order, if that's a confusing phrase, we're essentially asking this question in orange. Could you reconstruct a map from only the rank order of intercity distances? So the rank order being statements like Ottawa is closer to Toronto than Calgary is to Vancouver. So essentially a pairwise measure of dissimilarity or distances. And can we do that to make a 2-axed map? A map that has no units. The units don't matter right here, right? And it's essentially the constellation of the points of the sampling of the species within these sites. And I'm going to unpack the map and the distance analogy above the confused face and say, can we do this? We know what the map of Canada looks like, but consider the capital cities spread across the country. If we could figure out the rank order distances between these locations, could NMDS recreate their positions on a map? So what is the rank order distances? Well, it looks like the back, to an old person like me, it looks like the back of what the road atlas, the big map in the back of the car used to look like. So the distance of kilometers between 2 cities. So Ottawa is closer to Toronto than Calgary is to Vancouver. So can NMDS do this? And in the NMDS analysis, which is like 2 or 3 lines of code in your R routine, if we feed it this pairwise dissimilarity matrix or distance matrix, which is this, so that the cities and their pairwise distances that are in rank order, this is what we map. This is what it reduces to. Excuse me. So 2 axes where we see a far east city, a far west city, and actually a fair amount of longitudinal accuracy. Now we see us, and I think what's present in the data there is that most of our cities are arranged along not much latitudinal variation, right? But in this case, where we see latitudinal variation is within kind of Ontario and Quebec, and so that's inflated here on the second axis, that we've got kind of a flipped north-south here. So it does an okay job, and we can do that by then adding, as you would in some environmental data. So we're not going to use temperature data. We're not going to use the pH of the water, but those are things that scientists, ecologists would use with their kinds of NMDS analyses. But in this case, we'll use latitude and longitude as the metadata to see if we can put vectors on this NMDS to explain those variations, or to put provinces on. And you'll see in the first NMDS, which is the same kind of plot that we just saw, but we just colored it. And instead, in the colors we've applied are cities within the same province. And you see that Ontario clusters and Alberta clusters and then Quebec clusters, and we've got not so bad categorical capturing of the data. From just the pairwise distances, this is a map that you wouldn't want to drive by, but it's a map that if you kind of take your glasses off and squint at it, it's like, yeah, I see the shape of Canada there. And then we add our metadata and we say, oh yeah, okay, so there's the principal first NMDS axis is largely longitude, and the second one is largely latitude. So what other continuous variables might we want or might we have for the sites where we have where we're interested in using NMDS to ask ecological questions? So let's put aside, this is really the an analogy about what NMDS could do. So here it is. Let's look more specifically at a biological example to look at some of these parameters and say in Harvard forests there has been 8 long-term monitoring plots where over 12 years the diversity of ants were sampled using 4 different methods, and they were sampled 3 times a year. As you can see in pink overlaid on this topography, we've got different sites, some hardwood sites, some hemlock sites, and sites that have been logged, that's the L, or girdled, which is a anti-hemlock, a looper precaution. And in this scenario, the researchers, the entomologists, were interested in whether or not the plots are different for the component of ants that live there. And so you can see across the top here, this is what an incidence matrix would look like. And so we've got ant species and the sites, and zeros or ones where we see them. So we can look at this and kind of, even just kind of, again, taking the glasses and looking at it, we can see, well, some of these species, some of these sites seem to have more ones than others. Some of these ants seem to be more widely distributed than others, like these Sophienogaster. These 2 Sophienogaster species like living in forests, whatever the treatment. Whereas we see some of the other, like the Camponotus here, this carpenter ant that lives only in 1 or 2 locations. So the scenario is to take the observed incidence matrix and using different kind of dissimilarity calculations. You might have been introduced in 1070 years ago in your life, you might have been introduced to the Jacquard index or the Bray-Curtis index. There's a whole bunch of, there's a diversity of beta diversity indices. Now this Bray-Curtis measure is converted for each one of these sites, the presence and absence of these ants, to this dissimilarity number, which is essentially a pairwise distance matrix. The mathematics behind that you can unpack later on in other courses, but it's going to be useful for you. So Bray-Curtis, if you don't remember, is if you've got 2 sites, A and B, that share J species, and then the species only found at A and the species only found at B, that's roughly the Bray-Curtis calculation there. And it's bounded, it's an index between bounded and 1, so at 0, the 2 sites have the same composition, a dissimilarity matrix, and 1 means they don't share any. And so what the researchers with ants at Harvard Forest unpacked was they used that dissimilarity matrix to create this NMDS plot of the sites that were hardwood, in light blue, the hemlock sites, so that conifer in green, sites where the trees had been girdled to prevent the infestation, the looper infestation, or just the outright log. And so here they're essentially using that pairwise, the incidence matrix, to create the pairwise distance matrix to map this to help us visualize whether or not these communities are different. This is a powerful simplification of complicated data that let us answer the question of, are the 2 places, or the 4 places in this case, different or not? Now, there are statistics that are associated with this. One of them is a statistic called Permanova, or an analysis of similarity. They both the output essentially looks like an ANOVA, where we're comparing the variation within a site to the variation between all of these sites. And they'll give you an F value and a P value. And the appropriateness of an NMDS approach is something that we do using a called a stress plot. And one of the outputs of your R routine is going to create what's called a Shepherd diagram like this, which is the observed dissimilarity, so the thing that we calculated in nature, and then this ordination distance, so the simplification that is happening in the constructs of the calculations. Do they look like the dissimilarity observed versus look like the dissimilarity simplified? You can measure that with the R squared, as has happened there, but you can also do a sum of the squared residuals, which is called stress, which is what Lisa's going through. And it's an easy number to be fed to you in your R routine. And traditionally, if it's less than 0.2, it's generally useful. Less than 0.2 or 0.3. But then we can also talk about whether things are nested or whether things are, whether species is nested one within another or turnover. So this is the Harvard forest analogy we just talked about. And it kind of, those sites kind of move around. You see the discs there, if you're going to the eye doctor, which I clearly do, they're not clearly overlapping, right? There's your hemlocks that twist down slightly to your girdle, that twist down slightly to your hardwood, that rotate again, kind of counterclockwise there. So this would be more of an example of what's called a turnover. So the species in one site are not just a subset of the species at another site. If that's the case, that's a nested. And this other kind of ant-based analysis, this black rock forest here, is more nested. But a classic nested would just be one inside another, inside another. And so these are important ways to consider and to visualize beta diversity, because there's ultimately consequences if sites are threatened, either by development, like in our scenario, or by whatever kind of destructive forces might be at play. If they're nested, the sites, if there's turnover, losing one is essentially you've lost that complement of species. If they're nested, as long as you haven't lost the site that contains all of the greatest range of diversity, you might be okay. So in your InvertebrateR assignment, what you're going to do is take this NMDS approach. And again, I want you to use R. I want you to be familiar with R. I want you to be comfortable with R so that you can answer these questions about the deep sea thermal vent communities around the world. And there are questions that you will then answer in your video. Now, there's 2 other videos associated with the assignment. One of them is going through an example in R with the code that kind of lays out the code that you're going to use to make these kinds of conclusions for alpha and beta diversity. And the other video is thinking about how to present it in a really time-constrained perspective. So, this is the end of the beginning, not the beginning of the end. But welcome to your second InvertebrateR assignment. I hope you enjoy it, and I hope you appreciate the time that you get to spend with these special places and learning a little bit more and advocating for this special diversity. So, take care of yourself, take care of others around you, and enjoy.